Hello, everyone, and welcome to today's session. And today we'll be discussing the NCERT exemplar questions for the chapter Molecular Basis of Inheritance. Okay, this is the part 2 of the NCERT exemplar questions. Before starting the lecture, I just want to give you more information. The images, text, and other materials used in our presentations are of education purposes only. We respect the intellectual property rights of the original creators and owners. If you are the owner and have any concerns or issues, please contact us at sathee@itk.ac.in and we shall remove the content immediately. Please note that parts of the content may have been generated using generative AI, which is experimental. Okay, so what are the objectives of today's session? So today we will discuss the NCERT exemplar questions and we will understand the fundamental concepts which are related to this chapter. Okay, so you can see the questions have been divided into the following category. Okay, so we have multiple choice questions. Then we'll discuss very short answer type questions. We'll also discuss short answer type questions and long answer type questions. Okay, so let's begin with the session. First, let's talk about the multiple choice question. Okay, now let's read the question. The question is saying that which of the following step in transcription is catalyzed by RNA polymerase? Now, what was transcription? When RNA is formed from DNA, this is transcription. Okay. RNA polymerase is involved here. So RNA polymerase is a DNA dependent. Uh, okay. It is dependent on a DNA template. Okay. And it produces RNA, which is complementary to that template. So in which of these steps RNA polymerase is involved? So your options are initiation. Option number B is elongation. Option number C is termination. And option number D is all of these. So what will be the correct answer? Okay. So let's have a look at the answer. So answer is option number D, all of these. RNA polymerase is involved in all of these reactions. Okay, so transcription can be divided into 3 stages: initiation, elongation, and termination. Now, what happens during initiation? So RNA polymerase will bind to the promoter region. And this binding to the promoter region, this is, you can say, done with the help of sigma factor. So what is happening in the initiation step? Initially, you have the core RNA polymerase. So this core RNA polymerase cannot bind to the promoter. Okay, now comes the sigma factor. Sigma factor then binds to the core RNA polymerase. And now it is known as the holoenzyme. Now it is able to recognize the promoter with the help of the sigma factor. Okay. Then the transcription is initiated. Next, elongation step, RNA polymerase polymerizes the RNA chain in 5' to 3' direction. So this, remember this direction, 5' to 3'. Then next step is the termination step. Here the RNA polymerase will recognize the stop signals and with the help of the rho factor, what will happen? With the help of this rho factor, the RNA DNA hybrid which was being synthesized, this hybrid is broken down and RNA is separated from the DNA. Okay. So this rho factor, these are involved in the prokaryotes. Here you can see in this image, This is the RNA polymerase. This is synthesizing the RNA which is complementary to this template in 5' to 3' direction. Clear? Now let's move on to the next question. Next question is, the control of gene expression takes place at the level of. Okay, so you can control the gene expression at various levels. Okay, so you are given 4 options here. Okay, so you have to select the correct answer from these options. The first option is DNA replication, second is transcription, option number C is translation, and D is none of these. So what will be the correct answer? Okay, so let's have a look at the answer. Option number B is the correct answer, transcription. Okay, so let's have a look at the explanation. So control of gene expression primarily takes place at the level of transcription. So what is happening here? You can see this control of gene expression at the transcription level. This is the most common and efficient level of gene regulation. Now, it prevents unnecessary production of the messenger RNA. Okay. So, it saves the cellular energy as well as the resources. You can see only this mRNA is single stranded. Only one of the template is transcribed into RNA. Okay. So, if you are doing regulation at the transcription level, then you are saving the energy also, because unnecessary mRNA are not being produced. In producing RNA, resources are being utilized. Okay. So you can see this kind of gene control which is happening at the transcription level. This is happening, or this is occurring before the gene is expressed. What does this mean? So this means gene is expressed means proteins are being produced. So gene is transcribed into DNA is transcribed into RNA. RNA is translated into proteins. So before the synthesis of the proteins, the control of the gene expression is happening at the transcription level. Or you can say before translation. Or simply you can say before the gene is expressed into the proteins. Now, next question. So, regulatory proteins are the accessory proteins that interact with the RNA polymerase and affects its role in transcription. Now, which of the following statement is correct about the regulatory protein? So, what are the options? Option number A is they only increase the expression. Option number B, they only decrease the expression. Option number C, they interact with RNA polymerase but do not affect the expression. And option number D, they can act both as activators and as the repressors. So what would be the correct answer here? Okay, let's have a look at the answer. See, option number D is the correct answer here. They can act both as activators and the repressors. Now let's talk about the dual role of the regulatory proteins. So these proteins, they can function as activators also, means they are involved in the positive regulation. Okay, and they enhance the transcription. When they enhance the transcription, it is known as positive regulation. They can act as repressor also, means they are preventing the transcription. Okay, and they are doing the negative regulation. So same protein might act as both activator and repressor depending on the context. Okay. Now let's talk about this question. Which was the last human chromosome to be completely sequenced? You must have studied about the Human Genome Project. In Human Genome Project, human genome was sequenced. So they are asking which was the last chromosome to be completely sequenced. Options are chromosome 1, chromosome 21, chromosome 11 is the option number B, and option number D is chromosome X. What will be the correct answer? Okay, so let's have a look at the answer. The option number A is the correct answer, chromosome 1. Chromosome 1 was the last completed chromosome which was sequenced 2 decades after the Human Genome Project. Okay, it is the largest human genome as well, chromosome as well. Okay, that's why it was sequenced in the last. Next question, which of the following are the functions of the RNA? So RNA have different functions. Okay, so 4 options are given to you. You have to choose the correct answer. First option is RNA is a carrier of the genetic information from DNA to ribosome synthesizing polypeptides. Okay. Next option is it carries amino acid to the ribosomes. Option number C, it is constituent component of the ribosome, and option number D is all of the above. What would be the correct answer? Option number D, all of the above. This is the right answer. So let's talk about the functions of the RNA. The RNA is very good for the transmission of the information, and DNA is good for storing the information. Clear? So mRNA carries the genetic information which is stored in the DNA to the ribosomes, leading to the synthesis of the polypeptides, or simply you can say proteins. Now, why other options are incorrect? So, transfer RNA, it carries the amino acid to the ribosome for protein synthesis. Okay, transfer RNA acts as an adapter molecule between the genetic code and the amino acids. Now, ribosomal RNA, it acts as a structural unit of ribosomes along with the proteins. Okay, that's why answer is option number D, all of the above. So next question, this is a very simple numerical, you can say you have to solve it. Now, question is, while analyzing DNA of an organism, a total number of 5,386 nucleotides were found, of which the proportion of different bases were adenine was 29% in that DNA, guanine was 17%, cytosine was 32%, thymine was 17%. Now, considering the Chargaff rule, it can be concluded that. Okay, so simply in simple words, you can say what does Chargaff rule says? Number of adenine equal to number of thymine, number of guanine is equal to number of cytosine. Okay, or ratio of A by T is equal to ratio of G by C. So, what are the options? Option number A, it is double stranded circular DNA. Option number B, it is single stranded DNA. Option number C, it is double stranded linear DNA. Option number D is no conclusion can be drawn. What is the correct answer? So let's have a look at the answer. Option number B is the correct answer. It is single stranded DNA. So according to Chargaff rule of base pairing, the amount of adenine is always equal to amount of thymine, and amount of guanine is equal to amount of cytosine. Okay, A is joined to thymine with the help of 2 hydrogen bonds, and G is joined to cytosine with the help of 3 hydrogen bonds. Now, what does Targef rule says? The ratio of adenine to thymine, A by T, and that of guanine to cytosine is always equal to 1. Okay, so A by T is equal to G by C is equal to 1. Now, if you look at this information given in this question, A is 29. So T should also be 29, but here T is 17%, G is 17, but cytosine is 32. So G and C should be equal, A and T should be equal. But this is not observed here. So we can say this DNA is not following Chargaff rule because Chargaff rule is for double-stranded DNA. Okay, remember Chargaff rule is for double-stranded DNA. So here Chargaff rule is not being followed. So we can say, or we can conclude that it is single-stranded DNA, not double-stranded DNA. That's why option number B is the correct answer here. And not any other option. Clear? A, C or D. Now next question. If the sequence of nitrogen base is in the coding strand of DNA in the transcription unit is 5' ATGAATG 3'. The sequence of bases in the RNA transcript would be. So let's understand this question first. So they have given the sequence of the coding strand. Okay, what is the sequence of coding strand? This is the sequence of coding strand. Now you have to tell the sequence of the RNA which is formed. Okay, these are the 4 options. What will be the correct answer here? So let's have a look at the answer. See, option number A is the correct answer here. So what is happening? Okay, so remember this. The coding strand of the DNA, it has a similar sequence to the RNA or messenger RNA, you can say. So they both have same sequence. Okay, or you can say similar sequence. The only difference is that in RNA you have uracil instead of thymine. Okay, so DNA have thymine, uracil has RNA has uracil. This is the only difference. Otherwise, the sequence is same. So you can see this was the sequence of the coding strand. So replace this T with U because in RNA you have U instead of T. So your sequence will be A, U, G, A, U, and G, and from 5' to 3'. So this is the correct answer here. Clear? So this we have discussed. Now let's move on. Now, if the base sequence of a codon in mRNA is 5' AUG to 3', the sequence of transfer RNA pairing with it must be. So they are saying in messenger RNA, you have the codons. Codons are the triplet of the nucleotides. Okay, these codons are recognized by the transfer RNA. Transfer RNA has an anticodon loop. So if in the messenger RNA you have this sequence AUG, so what will be the sequence in the anticodon region of the transfer RNA? It is a very simple question. So options, these are the 4 options given to you, A, B, C, D. You have to choose the correct answer. So let's have a look at the answer. See, option B, 5' CAU is the correct answer. Okay. Remember the polarity. It is saying 5' to 3'. If you write it 3' to 5', it will be UAC. Now, according to base complementary rule, for adenine in messenger RNA, you have uracil in thymine instead of T. And for uracil in messenger RNA, the complementary base in transfer RNA will be A. Okay, but for G in mRNA, complementary base will be C. Clear? So you can see in messenger RNA, the sequence of codon is AUG, 5 prime to 3 prime. So what will be the sequence in the anticodon region? So A will be complementary with the U, then U with A, and G with C. Clear? So sequence will be, you can say UAC from 3 prime to 5 prime or CAU from 5 prime to 3 prime. So you have to do what? You have to just make the complementary of this sequence, AUG. Okay? And then you will get the answer. Here you can see this is a transfer RNA. Transfer RNA has an anticodon loop here. So AUG is the sequence. AUG is the codon in the messenger RNA. AUG codes for methionine. It is the initiator codon. Now transfer RNA recognizes the initiator codon, and the transfer RNA will have the complementary sequence to this codon. So sequence will be UAC from 3 prime to 5 prime, or you can write CAU from 5 prime to 3 prime. So next question is, the amino acid attaches to the transfer RNA at its... Okay, so transfer RNA brings the corresponding amino acid to the ribosomes. So where does the amino acid attaches? At what region does amino acid attaches to the transfer RNA? So what are the options here? Okay, 5 prime end, 3 prime end, anticodon site, or DHU loop. What is the correct answer? Okay, option number B, 3 prime to 3 prime end is the correct answer. Now 3 prime end of transfer RNA is also called the amino acid acceptor arm. So this amino acid binds to the 3 prime end of the transfer RNA. Okay. Now the 3 prime end of the transfer RNA ends with the sequence CAA. Remember this. Now this terminal adenosine is where the amino acid attaches. Okay, this is the terminal adenosine. So here you can see this is the 3 prime end of transfer RNA. Here the sequence will be CAA and the amino acid will bind to this A, this terminal A. Okay, this is clear. Now the anticodon site will pair with the codon present on the mRNA. One of the option was DHU loops. So DHU loops helps in maintaining the transfer RNA stability. 5 prime end is not involved in amino acid attachment. This is the 3 prime end. Okay. So here you can see this is the 5 prime end. This is the 3 prime end. Now next question, to initiate translation, the mRNA first binds to. Translation is synthesis of proteins from the mRNA. So here the mRNA will bind to options are smaller ribosomal subunit, larger ribosomal subunit, the whole ribosome, or if option number D is no such specificity exists. What is the correct answer? So let's have a look at the answer. C, option number A is the correct answer. Smaller ribosomal subunit. Clear? So what is happening here? The mRNA will first bind to the smaller ribosomal subunit to initiate the translation. Then what will happen? This small ribosomal subunit, this will scan the messenger RNA. Okay, and it will locate the initiator codon, which is AUG, and then the transfer RNA will also recognize this initiator codon AUG, and then the large subunit will come and the complex will be completed. Okay, the small ribosomal subunit contains the site where the mRNA binding occurs. So mRNA binds to the small ribosomal subunit. It helps in scanning the mRNA to locate the start codon. Okay, it ensures proper positioning of the mRNA for the translation as well. Here you can see this is the small ribosomal subunit. The small ribosomal subunit will bind to the messenger RNA. Okay, and this small ribosomal subunit scans the messenger RNA and it locates the initiation codon, which is AUG, and then the transfer RNA will come that will have an anticodon loop. Okay, this will bind here. Okay, and it will bring the corresponding amino acid, which is the methionine. So AUG codes for methionine. Next question, in E. coli, the ELAC operon gets switched on when? The options are when lactose is present and it binds to the repressor. Option number B, repressor binds to operator. Option number C, RNA polymerase bind to operator. And D, lactose is present and it binds to RNA polymerase. What is the correct answer? So let's have a look at the answer. The option number A, lactose is present and it binds to the repressor. So lactose acts as the inducer of the lac operon. So what happens in the presence of the lactose? So repressor protein, which is being produced by the regulatory gene, this repressor proteins bind to the, okay, this repressor protein usually bind to the operator. But what happens in the presence of the lactose? This lactose will bind to the repressor. It was called conformation change in the repressor, and now the repressor cannot bind to the operator region. Because of this, now RNA polymerase has access to the promoter and it can bind to the promoter, and the transcription is initiated now. Okay, clear? So in this case, lacopron is said to be switched on. So you can see this is the regulatory gene. This regulatory gene is producing repressor protein. This repressor protein, this becomes inactive repressor in the presence of the lactose. So lactose binds to this repressor protein. Okay, it causes conformation change in the repressor protein. And now this repressor cannot bind to the operator. And now RNA polymerase can bind to the promoter. And the transcription will be initiated. So mRNA will be produced, and from the mRNA, proteins will be produced. Okay, so there are three structural genes in the lac operon. You can see Z, Y, and A. Z code for beta galactosidase, Y code for permease, and A codes for trans-acetylase. Okay. So when the lactose is present, we know what will happen. It will be lac operon will be switched on. Transcription will occur. Clear? Now, in the absence of lactose, what happens? The repressor can bind to the operator, and the RNA polymerase now cannot bind to the promoter now, and thus the transcription is inhibited. Okay. So here you can see one of the option was wrong, because when lactose is present, repressor will bind to the lactose and now it cannot bind to the operator. One of the option was RNA polymerase bind to operator. This is also wrong because RNA polymerase bind to the promoter, not operator. Lactose bind to RNA polymerase. This is also wrong because lactose binds to the repressor, not RNA polymerase. Okay, this inducer is lactose. Lactose is binding to the repressor. This is a repressor protein. The next question is based on your understanding of genetic code. Explain the formation of any abnormal hemoglobin molecule. What are the known consequences of such changes? Okay, this is a very short answer type question. How do abnormal hemoglobin molecules are formed? So let's have a look at the answer. Okay, so abnormal hemoglobin is formed due to mutations in the DNA sequences. So these mutations will cause the change in the DNA sequence. Okay. The most common example is sickle cell hemoglobin. So what happens in sickle cell anemia? There is a mutation at the 6th position of the beta globin chain. Okay. And this is a single nucleotide polymorphism. Means there is a change in the only one base pair. Okay. Due to this change in only one base pair, the codon is changed and this codon now codes for some another amino acid. So usually in the normal hemoglobin, this codon codes for glutamic acid, but in the case of sickle cell anemia, it codes for valine. So here there is a single nucleotide polymorphism, or you can say there is a point mutation. Okay, it is occurring at a one point only, and there is change in only one base pair. So in the normal hemoglobin, you have GAG in the normal hemoglobin gene, and in the case of hemoglobin, this sickle cell anemia gene, you have GTG. So instead of A, you have T now. So this is point mutation occurring at only one point. It is also known as single nucleotide polymorphism. So in the normal hemoglobin, you have glutamic acid. This is replaced by valine. You can see normal RBCs. Normal RBCs are biconcave. So this is the codon sequence, GAG. Okay, in case of sickle cell RBCs or sickle cell hemoglobin, you have GTG instead of GAG. So instead of A, you have T here at the second position of the codon. Okay. So the protein which will be synthesized here will consist in the normal hemoglobin, you have glutamic acid. In the sickle cell hemoglobin, you have valine at the 6th position. So there is mutation at the 6th position of the beta globin chain. So hemoglobin, it is made up of alpha globin chain and beta globin chains. Here there is a mutation in the 6th position of the beta globin chain. At the 6th position, this point mutation is happening. Okay. So instead of A, you have T here. Okay, now the RBCs become sickle shaped, you can see here. Now, what are the consequences of sickle cell hemoglobin? The RBCs will become sickle shaped from biconcave shaped. What are the clinical effects? Now, as they are sickle shaped, they are not able to carry oxygen. So, there will be reduced oxygen carrying capacity leading to anemia. There will be altered RBCs. Okay, these altered RBCs, they cannot pass through narrow capillaries. As a result, blood capillaries are clogged also and thus affect blood supply to different organs as well. So you can see this can be quite dangerous also. Next question is, sometimes you can see cattle or even human beings give birth to young ones that are extremely different set of organs. Okay, like limb or position of the eye, you have to comment on this statement. And for more information, you can read NCERT section 8.5.8 as well. Okay, so this is dependent on the genes. Okay, there is a disruption of the gene expression during the development. This leads to the formation of this different set of organs. Okay, so everything is dependent on the expression of the genes. So organ development is a very complex process which are controlled by the genes. Okay? These genes must be expressed in a coordinated manner. If there is disruption, okay, of this gene expression, then you can see what are the consequences. The young ones can have different, extremely different set of organs. Clear? So sometimes there can be mutations, or due to environment, these disruption can occur, which will disrupt the gene expression. Okay, so let's have a look at the answer. So these birth defects, so these are known as birth defects. So they are by birth, they are known as congenital. Okay, so these birth defects are caused by disturbance in the coordinated regulation of the gene expression during the development. Remember this. Okay, gene expression is being disturbed here. Now, this coordinated regulation of the gene expression is very essential for proper organ formation and placement. Okay. Now, this disruption can occur due to genetic mutation or due to environmental influences. Clear? Next question. Now, in the nucleus, the number of ribonucleotide triphosphate is 10 times the number of deoxyribonucleoside triphosphate, but only deoxyribonucleotides are added during the DNA replication. You have to suggest a mechanism for this. So let's have a look at the answer. Okay. So you can see, so DNA polymerase that will only add nucleotides. Okay. It can only add nucleotides at the 3' end of the primers. So that's why they are used. Now, next question, name a few enzymes involved in DNA replication other than DNA polymerase and ligase, and you name the key function of each of them. Okay, this is a very straightforward question. Let's have a look at the answer. So, enzyme involved in DNA replication other than DNA polymerase and ligase are, first one is helicase. So, helicase will open the helix. Okay. So, first of all, the unwinding of DNA is required. Okay. So, DNA is double stranded. Okay. So, it has to be opened. This is known as unwinding of the DNA. This is done by the helicase enzyme. Clear? So, we require a single stranded template for carrying out the transcription. Okay. One is the leading strand, another one is the lagging strand. So, helicase will open the double helix. Then the primers can bind. Then DNA polymerase can add this nucleotide to the 3' end of the 3' OH of the primers. Next is topoisomerase. So it removes the supercoiling of the DNA. So when the DNA is being unwinded by helicase, so some tension is created. This tension is removed by topoisomerase. It removes the supercoiling of the DNA. And then you have the primase enzyme. This synthesizes the RNA primers. So DNA polymerase require RNA primers to synthesize the RNA. But RNA polymerases do not require any primers. Remember this is a very major difference between DNA polymerase and RNA polymerase. Now you have telomerase also. This synthesizes the DNA at the telomeric end of the chromosome. Okay, now next question. You have to name 3 viruses which have RNA as the genetic material. Okay, viruses which have RNA as a genetic material, they are known as retroviruses. Again, this is a very simple question. Usually plant viruses are RNA viruses. Some of the animal viruses or some of the viruses which cause disease in humans, they also consist of RNA. Okay, so what is the answer? See, some viruses can have RNA as a genetic material as well. Okay, such as tobacco mosaic virus. It is a plant virus. See. It consists of RNA. Then QB bacteriophages, they have RNA. HIV, human immunodeficiency virus, which causes AIDS. This also have RNA. Then influenza virus. So these all viruses you can see they have RNA as a genetic material. They are retrovirus. You can see this image. This is Q beta virus. Here you can see it has single-stranded RNA as a genetic material. Okay. Then you have this tobacco mosaic virus. Again, you can see it has RNA as a genetic material. Okay, so usually viruses, they are nucleoproteins. They have the nucleic acid also, and they have the capsid, which is made up of protein. That's why viruses, they are called nucleoproteins. And they are acellular. They are not cellular beings. They are acellular. Okay, now next question. Again, this is a very important question you can see. Now the question is saying a low level of expression of lacoperon is happening all the time. What does this mean? Okay, means it is being expressed constitutively. And what is the reason for that? Okay, can you explain the logic behind this phenomena? Okay, let's have a look at the answer. Okay, for more information, you could read NCERT section 5.8.1 as well. Clear? See, the low level of expression occurs all the time. So this is crucial because it ensures that a small amount of the enzyme needed to metabolize lactose is always available. So lactose is metabolized by the enzyme beta galactosidase. Okay, it breaks down lactose into galactose and glucose. Okay, so this basal level expression allows the cell to quickly respond to the presence of the lactose. Okay, clear? And one more thing, this lac-operon consists of an structural gene which codes for permease. So permease is required for what? So permease is coded by the LAG-Y gene. Okay, and this permease helps in the transport of lactose into the cell when lactose is available. So see, when lactose is added in the media or when the lactose is present, then it has to enter into the cells. How it will enter into the cells? With the help of permease. So we need permease all the time. Okay, if permease is not present, the lactose will not enter into the cell and it will not be metabolized. Okay, that's why the lac-operon is constitutively expressed. It is expressed all the time, although at a very low, you can say low rate. Okay, so why? Because permease is being produced and permease helps in the internalization of the lactose. Means for transport of the lactose into the cell, you require permease and then only the lactose will be metabolized. The next question, the total number of genes in human is far less, 25,000 than the previous estimate of up to 14,000 gene. You have to comment. So reason why less number of genes. Next is one of the reason is alternate splicing also. So what happens in alternate splicing? See, during alternate splicing, the exons can be mixed and matched again. So eukaryote, they have split genes. You have exons, then introns, exons, then introns, then exons, then introns. These introns are removed, then exons are joined together, and exons are translated into proteins. So what is happening in alternate splicing? Different combinations of exons are being produced. Okay, so you have exon 1, 2, 3, 4, 5, 6. This can be one sequence. Then you can have exon 1, then exon 3, then exon 2, then exon 5, then exon 4. So these exons, they are mixed together, and different, you can say, sequences are being produced, and these different variants, you can say, they produce different proteins. Clear? So this eliminates the need for separate genes for each protein. So we do not require separate genes for each protein. So because of this alternate splicing, different combinations of the exons are being produced and different proteins are being produced. Okay, by the same gene. So you can see here, these are 4 exons. Now, different combinations are being made. You can see this is the purple one, this is the blue one, then you have the yellow one. Here in second case, you have this purple one, then you have the green one, and then you have the yellow one. Now, after translation, they are producing 2 different proteins. So one gene is producing many proteins. So you do not require separate genes for each protein. That's why you have less number of genes now as compared to the earlier estimated number of 1,40,000 genes. And out of these, human DNA, only 2%, less than 2% of the DNA codes for the proteins. Okay. Next is post translation modification. So, translation means production of the proteins. So, there are some post translation modification means after the translation. So, it occurs after the protein is being synthesized. Now, in this modification, chemical changes are made to the amino acids. Okay, example, the protein can be phosphorylated, phosphate group can be added. It can be, you can say, glycosylation is one process, and ubiquination is one process. Where ubiquinone can be added. Here the glycosyl group is being added. Okay, so this increases the functional diversity among the proteins. So you have the same proteins, but different groups are being added to these proteins. Now they perform different functions. Okay, so now again one gene, there is only one gene, and you have multiple proteins now. Okay, clear? In alternating splicing, you have multiple proteins, but here you have a single protein, but this single protein is being chemically modified. So you can see these are proteins which are produced. Now disulfide bond can be added, then hydroxylation can be done, ubiquination can be done. Okay, methylation can be done, methyl group can be added, acetyl group can be added, which is acetylation, phosphorus group can be added, which is phosphorylation. Okay, then you have somolylation, lipidation. Okay, so the proteins now will perform different functions. The protein is same, the sequence is same, only the functional group is different, or the group is different, which has been added to the protein. Now, next question, the background information did Watson and Crick have made available for developing a model of DNA? What was their contribution? Okay, what background information did Watson and Crick have? Okay, that were used for developing the model of the DNA. So, what model of DNA they gave? They gave the double helical model of the DNA. Okay, so let's have a look at the answer. See, Watson and Crick had the following information which helped them to develop a model of DNA. The first one is X-ray diffraction data. So Rosalind Franklin high quality X-ray diffraction images were available to Watson and Crick. Then Morris Williams X-ray crystallographic data was available to Watson and Crick. Now these showed DNA had a regular repeating structure and helped determine its dimensions. इस मैं showing you Watson and Crick with their DNA model. Okay, next is the chemical composition. So ergin chargaff rule showing equal ratio of A by T and G by C bases. Okay. So now we know that A pairs with T and G pair with C. Clear? So that means there are two strands which are complementary to each other. Okay. Number of A is equal to number of T and number of G is equal to number of C. Okay. This is clear. Now Watson and Crick contributions were, what were their contributions? The first is correctly interpreting all available data to propose the double helix model. Okay, so they interpreted all the data which were available to them and they proposed the double helix model. So they said the DNA consists of two helical strands which are running in opposite direction. Okay, means the polarity is different. Okay, one is from 5 prime to 3 prime, another is from 3 prime to 5 prime, means they are anti-parallel to each other. Okay, the DNA have a sugar phosphate backbone. Base pairs are on the inside, so nitrogenous bases are towards the inside and they are forming pairs with each other. A pairs with T and G pair with C. Okay, this is clear. Now next question, what are the functions of methylated guanine cap and poly A tail in the mature RNA? So the RNA is processed. Okay. Now at the 5 prime end, a methyl guanine cap is being added, and at the 3 prime end, poly A tail is added. Now what are the function of these modification that is happening? So let's have a look at the answer. What are the function of methylated guanine cap? The first one is protection. So this methylated guanine cap, this protects the mRNA from 5 prime to 3 prime exonuclease degradation. So after the synthesis of RNA, the RNA is transported into the cytoplasm in case of eukaryotes. Okay, so this mRNA is protected from the 5 prime to 3 prime exonuclease degradation. Okay, it increases the mRNA stability, and then it helps in the recognition by translation initiation factors also, because mRNA is further translated into the proteins. Here, so you can see this methylated guanine cap. Okay, it is protecting the mRNA also. It is increasing the stability of the mRNA also. So this mRNA is being actually tagged by this methyl guanosine cap. So it is tagging it. Okay, and it is asking that don't destroy it. It is increasing the stability also. And when this tag is present, methyl guanine cap at the 5 prime end, then it is recognized by the translation initiation factors. And then the translation will begin. It also helps, or it also facilitates binding of smaller subunit of ribosome to the mRNA. Okay, so you can see at the 5 prime end you have this methylated guanine cap. This is methyl guanosine triphosphate at the 5 prime end. At the 3 prime end you have poly A tail. Okay, then what is the significance of poly A tail? It also protects the mRNA, okay, from 5 prime to 3 prime degradation by the exonucleases, and increases the mRNA stability and half-life. Okay, you can see this is the poly A tail which is being added at the 3 prime end of the messenger RNA. Okay. Okay, next question. You have to define operon and you have to give an example and you have to explain an inducible operon. So let's quickly have a look at the answer of this question. So operon, so concept of operon was proposed by 1961 by Jacob and Monod. So operon is a unit of prokaryotic gene expression. These are present in prokaryotes such as bacteria. Now it includes structural genes, and it includes control animal elements also. Okay, so here is the image showing you operon. So here you can see it has a promoter, it has an operator. These are the structural genes A, B, C, D, and then you have the regulatory region also. This is the regulatory gene. Okay, so what are the constituent of operator? What are the constituent of the promoter? Structural genes. Then you have the promoter, operator, and the regulatory genes as well. Okay, so operon components, structural genes. So these are the DNA segment that codes for the messenger RNA, and then messenger RNA will be translated into proteins. Then you have the promoter region where RNA polymerase will bind. Operator region, this is the DNA sequence where repressor proteins will bind. And then you have the regulatory gene which codes for these repressor proteins. Okay, and this repressor protein will control the transcription. Then inducer is a molecule that allows the transcription by preventing the repressor binding to the operator. Okay, then let's talk about the inducible operon system. Okay, it is a regulated unit of genetic material which is switched on in the presence of a chemical. That's why it is known as inducible operon. Example is lactose operon in E. coli. So in the presence of lactose, what happens? Lactose will bind to the repressor. It will cause conformation change in the repressor. Now the repressor cannot bind to the operator. And now the RNA polymerase can bind to the promoter and it will start the transcription. And the lac operon is switched on, and the three structural genes Y, Z, Y, and A, they will be expressed and they will produce the proteins. So lac operon is example of inducible operon when lactose is present. So in the presence of lactose, the lac operon is switched on. These are the three genes, structural genes. Z gene codes for beta galactosidase, Y gene codes for permease, A gene codes for transacetylase. Okay, beta galactosidase gene, this hydrolyzes lactose into galactose and glucose. Permease, this helps in the internalization of the lactose into the cells. And transacetylase, this adds an acetyl group from the acetyl coenzyme A to the galactosides. Okay, so this is an lac operon is an example of inducible operon. So when lactose is absent, then it is switched off because now repressor can bind to the operator. When the lactose is present, repressor cannot bind to the operator region. Okay, this is an image showing you lac operon. So you can see in the presence of inducer, which is lactose, this is binding to the inducer is binding to the repressor protein. Now you can see this is the inactive repressor. This cannot bind to the operator. And RNA polymerase can bind to the promoter and it will initiate the transcription, and then these proteins will be translated. Messenger RNA will be translated into proteins. These three enzymes will be produced. In the absence of the inducer, the repressor can bind to the operator. RNA polymerase now cannot bind to the promoter, and there will be no transcription of the lac operon. Okay. So with this, we end this session. I hope this session was quite knowledgeable for you. Now we'll meet you in the next session. Thank you very much for listening.